Okay. Welcome back everyone to the BISV High School Usable Club. Today we'll be covering Campbell's chapters 44 through 47, I believe. So these cover a lot of physiology concepts, the first of which is osmoregulation and excretion. So osmoregulation and excretion for us is the job of our kidneys, right? What is osmoregulation? So this, you can think of osmoregulation as maintaining the balance of salt concentrations of different solutes within your body. You don't want too much sodium, you don't want too much calcium. Um, and what your kidneys do is they can choose what to put into the urine and what to take out of the urine and in that way they can affect the concentration of different things in your blood supply and therefore your entire body. Their animals fall into two categories, osmoconformers and osmoregulators. An osmoconformer maintains the same osmotic balance as their environment. So if the environment has a certain chlorine concentration, the animal's body, their interstitial fluid, their cells will also, not necessarily their cells, but their interstitial fluid will have a similar concentration to their surroundings. They're osmoconformers. They take on the concentration of their environment. They don't really change their own. The other category, we are osmoregulators, right? An osmoregulator is an animal that will actively change its its internal concentrations regardless of what the environment is like. You can kind of think of this as like cold-blooded versus. I don't think those are like biologically correct terms. You can think of these uh as just think of them as exothermic and endothermic. Yeah. And hydrobiosis. This is life without water. Um, and this is talking, I guess, in relation to this slide, it's talking about the importance of water. Nephron. So, transport epithelia. These are in your kidney. There are filtration units called nephrons, and these nephrons have certain transport epithelia. And these are cell layers that help to control what goes into and out of the urine. So this is what actually does these transport epithelia is what actually does the filtration. And one interesting thing that you should note is that these transport epithelia, they're simple or columnar cuboidal cell types. So all of them are like cuboidal in shape. They can either be one layer of cuboidal cells or multiple layers of cuboidal cells. But just a general rule of thumb, if a cell type has a excretion function or if a cell type has a function of excretion, there's a good chance it's cuboidal. And here we talk about getting rid of waste. So one waste of physiological importance is nitrogen, mostly because of how prevalent it is in our bodies, how important it is. It's found in all macromolecules. It's found in all proteins and all nucleic acids. So two of the four very important classes of macromolecules contain nitrogen, and metabolizing these proteins or nucleic acids produce nitrogenous wastes. And different animals, if the nitrogen builds up in your systems, it can like a lot of things, it can be dangerous, and different animals dispose of this nitrogen in different ways, right? Some animals like fish will just dispose of it like as ammonia because they can get it to diffuse out. Others, like we excrete nitrogen we excrete nitrogenous waste as urea, which is a slightly more concentrated form of ammonia. And then some birds and other animals, they really compact their nitrogenous waste into something. Hold on, let me just check that I'm recording. I am. Okay. They really concentrate their nitrogenous waste into something called uric acid. And this is because uric acid, I think it's easier to dispose of, but the drawbacks of like anything in life, in biology, there are pros and cons. Uric acid is, you have to take amino groups and concentrate them into this nucleotide looking thing, this amino ring. And that is energy consuming. That's very expensive. And that's why the way that the wastes are excreted depends on diet or energy. There are pros and cons to each. Uric acid, yeah, energy. Ammonia is hard to get rid of. Okay, so now let's talk about the human kidney, I guess. There are many different kinds of filtration systems in nature. This diagram on the left, this is a filtration unit of a human kidney, right? So you can see blood enters into, I'll just walk you through this. Blood enters from a renal artery into the glomerulus, which is, it's like a specialized set of capillaries that the solids within those capillaries are pushed into the Bowman's capsule. Those solids are channeled down the proximal tubule, into the descent into the loop of Henle, then up through the distal tubule, and then into what are these called? Into a collecting duct. And from here, all of the waste will be excreted through the through renal pelvi ultimately into your bladder, right? So from the glomerulus, these capillaries, you start off with like the base filtrate that is a representation of your blood. So the liquid part of your blood is basically the liquid part of your blood minus the proteins, minus the blood cells, is what gets excreted from the capillaries into the proximal tubule. And as that fluid moves through the nephron, there are certain parts that have specific functions that alters, that like puts in things to get excreted. And then maybe if there's glucose or if there's water and salts within the urine that you want to retain in your body, the nephron will reabsorb those. So as the solute travels through the nephron, different processes happen that change the composition. Filtration, reabsorption. These are the four functions of the kidneys. So the first is to filter your blood to get rid of things you don't want, right? That happens when solids are pushed into the nephron itself. Reabsorption, secretion. So reabsorption, you're taking back things that you don't want to pee out. Secretion, you're putting things in your pee that you want to get rid of, right? So you reabsorb good things, you secrete like wastes. And excretion is like like the peeing part, invertebrates. Kidneys will help with getting producing the urine itself, which is its excretion, and osmoregulation, where it can affect the solute concentrations in your body. Right. Okay. So now let's talk about the parts of the nephron again. Remember the afferent artery goes into the glomerulus. The efferent was I forgot which one was which. But anyway, here the first step along the way is the proximal tubule, and as you can see here, you hear. Okay. One thing that I want to clarify is that some things in the kidney, some things your kidneys can choose to reabsorb or excrete, right? Other things happen automatically. I think, regardless of regardless of what the kidney modifies, 80% of the 80% of the water and salts within the original filtrate get reabsorbed into your blood. That's not to say that that it's passively diffusing. No, the kidney is actively pumping salt and water back into your blood. It's just that regardless of external hormones, regardless of whatever signals are acting on the kidneys, 80% of water and salt gets reabsorbed into your bloodstream. Now, for example, if you've heard of ADH, antidiuretic hormone, what those hormones act on is the remaining 20%, right? So here in the proximal tubule, salt and water get reabsorbed. That's automatic. Nutrients get reabsorbed. If there's glucose here, which there shouldn't be. Oh, sorry. There is glucose here. And usually your kidneys can reabsorb all of it. And depending on the pH balance of your blood, it can either secrete hydrogen ions into the urine or take out bicarb. So here you can see how the proximal tubule can help regulate the pH balance of your blood, along with osmoregulation because of the salts and water. Also, here you see the ammonia. You're secreting the ammonia into the filtrate. Next, you have the loop of Henle, and this is a very powerful. It's a very powerful tool for reabsorption. So, basically, what this section does is, as you go down, it pumps a lot of salt into the surroundings, such that as up here, there's I think 400 concentration of solutes in general. Osmolarity is around 400. Down here, it's 1600. So there are 4 times as much solute in the inner medulla than in the cortex. And what does that mean? Well, if you have the filtrate that is going through the proximal, and then it starts ascending through the loop of Henle, as it goes down, the surroundings will become more and more concentrated relative to the inside. As a result, water will keep on leaving the tubule because of osmosis. So basically, as it moves down, water keeps moving out through osmosis, even though that makes the inside more concentrated. So by having this gradient of osmolarity, you're making the filtrate, you're concentrating the filtrate a lot. And then as you go up, the surroundings will now be much less concentrated, the surroundings will be less concentrated, so salt will be able to diffuse out like through channels. And once you reach like the upper half of the loop of Henle, this is called the thick ascending limb. And at this point, you actively pump salts out of the nephron, and this is what helps to establish the gradient of concentration, ion concentrations. I wish I could go into more detail, but just know that as you go down, the concentration in the outside decreases, so water leaves. As you go up, concentration of the solutes of the outside is less. The concentration of solids on the outside is less, so salt will diffuse out, and then in the ascending limb, you pump out salts actively. Right. And now the distal tubule, this is the source for a lot of like action via hormones, via one main thing is that there's a potassium and hydrogen, I think, co-porter, or sodium, potassium, hydrogen. One of those, you can either selectively transfer potassium or hydrogen ions. So there's a system that allows you to get one but not the other. So sometimes this has medical significance. And then a bunch of things, and then down renal, and then you go after so it goes proximal, loop of Henle, then to distal, then through the collecting duct. Okay. Now let's look at some of the hormonal control over the kidneys. One, there are two main hormones that control how the kidneys function, and one is produced in the posterior pituitary. This creates a molecule or a hormone called antidiuretic hormone, and what this does is ADH will cause you, so diuresis means peeing more. So antidiuretic means that you're reabsorbing more water and you're peeing less, right? And now when your hypothalamus detects that your blood has low levels of water and solutes, it will secrete ADH, which will cause this acts on the collecting ducts, and it secretes ADH, which causes the collecting duct to take up water and different salts. Then we have the juxtaglomerular apparatus, and this is a system that it is produced in the kidneys, it goes somewhere else, and then it comes back to the kidneys. So when there's a part of a blood vessel called the juxtaglomerular apparatus that detects low solute, low filtrate flow through the distal tubule, right? When it does that, it releases something called renin. And when it detects low solute flow, that means that you don't have enough water and salts in your blood, and therefore you need to reabsorb more. So it releases something called renin, which becomes catalyzed to angiotensin 1, and then that goes to the lungs and becomes catalyzed to angiotensin 2. Angiotensin 2 comes back to the adrenals, which sit on top of the kidneys, to tell them to produce aldosterone, which cause an increase in sodium and water reabsorption. And atrial natriuretic peptide I'm not sure how. Oh, um, I don't think this has to do with the juxtaglomerular apparatus, but the ANP is a hormone that tells the kidneys to secrete more sodium into the blood. Sorry, into the filtrate. Um, right. So this is produced in the heart, actually. Um, And basically, when you have a lot of blood volume, that means you have a lot of water and a lot of sodium, a lot of salts, right? And that increase in blood volume will kind of stretch the walls of the artery, of the atrium. And when the walls of the atrium are stretched, it will cause the heart to produce ANP. ANP will cause the kidneys, in turn, to secrete water and salts into the. It will cause the kidneys to secrete water and salts into the filtrate. As water and salts leave the body through urine, the blood volume will go down, and the heart, the stretching of the atria will decrease, and ANP will stop being released. So these are two examples of kind of negative feedback that control the kidney. Oh, okay, chapter 45. Now let's talk about the hormones and the endocrine system. Hormones are defined as small signaling molecules that are present in small quantities that act like by binding to a receptor. Right? So there are three types of signaling, namely paracrine, autocrine, and endocrine signaling. Paracrine signaling is you are releasing a hormone that diffuses to nearby cells, and it has an effect on those nearby cells. Oh, sorry. Another definition of a hormone is that it has to move through blood, I think. Correct me if I'm wrong. But paracrine, it releases hormones, it releases molecules that uh, that have effects on nearby cells. Autocrine signaling is like the stomach secreting gastrin, right? It secretes into the bloodstream, it circulates around, and then it goes and acts back on the stomach. So autocrine, a cell secretes something that acts on itself. And endocrine is what we think of when we think of hormones. You're secreting something in the bloodstream that has a widespread physiological effect. So the molecules travel all over the place. And yeah, classes of hormones, gases, polypeptides, steroids, amines. So these are some of basically the molecular classes that a hormone can belong to. So some gases like nitric oxide, I think it's a neurotransmitter actually, but yeah, there are hormones that are gases. Some polypeptides like what was it called? Angiotensin 2, aldosterone, maybe not aldosterone, but angiotensin 2 is an example of a polypeptide hormone. An example of steroids like cortisol, glucocorticoids, amines like epinephrine, norepinephrine. And the entire endocrine system, what's known as like the master gland, is the anterior pituitary. And it's called the master gland because it secretes a lot of stimulating hormone. And these stimulating hormones basically tell other endocrine glands, hey, you should grow and secrete more hormones. And the pituitary in turn is regulated by the hypothalamus. If you recall, the pituitary is right below the hypothalamus, And the hypothalamus has direct blood vessels, portal veins into the pituitary. So that's how they control it. More hormones, thyroid hormones. So let's just discuss some of the different important hormones. Thyroid hormones include T3 and T4. If I'm not mistaken, T3 is the metabolically active one. That means that T3 does the actual functioning, which in this case, thyroid hormone raises your metabolism. So it acts with vitamin D to as a transcription factor to up your metabolism, right? Parathyroid, the parathyroid gland is so these are like, I don't know if you can see on this, but right here, you can see the shape of the thyroid. These are like spots on the four corners of the thyroid. And this secretes a hormone called parathyroid hormone. And this is very important because it raises blood calcium levels. Calcium is an important neurotransmitter. It's an important ion for many different systems. I mean, you can think of it like all cells have a sarcoplasmic reticulum, and that's where you store calcium. So parathyroid hormone is very important. The adrenal hormones. So let's talk about the adrenal gland. Okay, the adrenal glands, as pictured here, consists of the adrenal cortex, which is the outside. In general, if you hear cortex, like you should think outside, like that holds in here, in cell biology, in plants. Cortex is the outside, right? The cortex produces hormones like mineralocorticoids and glucocorticoids. Mineralocorticoids are like aldosterone. They act on the kidneys to affect how much salt and water are excreted through your urine. Glucocorticoids are actually more interesting. They cause glucose release into the bloodstream, and they suppress your immune system. Now, this is one interesting thing is that chronic stress will trigger for excess production or continued production of glucocorticoids. Because of the glucocorticoids, stress can actually physiologically depress your immune system. And this is, I guess, one way that being stressed can lead you to be sick more often. Yeah. Now, also the cortex does produce some, like a little bit of sex hormones. So some aldosterone, some androgens, estradiol. And the sex hormones are mainly produced in the reproductive organs, like testes, ovaries. They lead to the secondary sexual characteristics and sexual dimorphism. Okay, now let's move to animal reproduction. Okay, so here are some examples of asexual reproduction. You have budding, which is, if you've seen hydras, it's a parent plant. It's a new organism will grow on a parent plant, and once it gets to a certain stage, once it gets to a certain phase, it'll like pinch off and become its own. It become independent. Right? Fission is, if you heard of binary fission, it's like cell division, except it results in new organisms, right? Because you create new individuals. That is known as reproduction. Fragmentation, this one's pretty fun. If there are certain animals like planarians, they possess the ability to self-regenerate from many parts. Like sea stars are probably a more well-known example. But if you cut off a planarian, they have an abundance of stem cells that will allow them to like if you cut the planarian in half, the tail half will regenerate a head, the head half will regenerate a tail. And this is technically reproducing because you're producing two new organisms, right? Or one new organism. Finally, parthenogenesis this is a weird one where you have an egg, like you have a female gamete. Normally during sexual reproduction and fertilization, like for humans, the eggs metabolism and like for the egg to start developing into a zygote, it has to get like kickstarted by usually by fertilization entry of the sperm nucleus. However, in parthenogenesis, the egg doesn't, it just goes. Like you don't, there's no sperm, there's no nothing. The parthenogenic animals generally, the population is generally all female, and their eggs just spontaneously activate and develop into new organisms. Hermaphrodites. So humans have one biological sex. You either have testes or ovaries, right? There are certain animals that have both testes and ovaries, or both sets of reproductive structures. And like snails, for example, they usually or earthworms, they usually like cross-fertilize, meaning they exchange sperm and then their sets of eggs are both fertilized. Yeah. Sex reversal. So this is, I think there's a specific type of fish, a reef fish, that I think some kind of parrotfish, that the largest if you have a group of females, the largest female will transform into a male. For example, if you these fish form, it can help to think of them as like a lion pride, where the male is the lion, right? The male fish is the lion. Now, if something happens to the lion, the male fish, the largest female will develop characteristics. The largest female will become a male. Yeah. I actually didn't know that about I actually didn't know that about clownfish. Cool. The more you know, right? Um, yeah. Now let's talk about what fertilization is. So this is a process where a basically the sperm nucleus enters the egg and activates it. It can be either external or internal. Humans have internal fertilization, but other animals have external fertilization where they release their gametes into the environment, right? Like some fish, frogs, they just like they just release everything into the water. Two things to note. First, if you have external fertilization, because of the likelihood that a lot of the gametes will be lost and damaged or destroyed, generally there are a lot more gametes released, right? Second thing is in a water with a lot of animals spawning, you don't want your sperm to go and fertilize the sperm of a different species. You don't want your sperm to go fertilize the egg of a different species, right? You want your sperm, you want to mate specifically with your own species. And so for these um all for these animals that spawn, it makes sense for them to have specific like very specific features that um ensure that only the right sperm can fertilize the eggs. And I think there are certain things that all animals, there are certain blocks to fertilization that all animals have. But for these animals, for these species, it makes sense to have more. Like, I was gonna say something, but uh, if I decided against it. Mhm. Okay, I'm sorry. The space gets me every time. The male. So where do I even start? Okay, so let's just I'll just give you a overview of this. So these are the testes. This is the epididymis. So in the testes, the germ cells begin developing into sperm. Um, the sperm then finish their maturation in the epididymis, um, and then uh, yeah, they finish their development through the epididymis, and when ejaculation happens, they go through this duct called the vas deferens. Um, and this is, if you've ever heard of a vasectomy, it's like the surgical resection of this tube. So if you have a vasectomy, the sperm can't reach the they can't go past the epididymis, and therefore you are I don't know, neutered? No. Therefore you don't have sperm basically. You don't release sperm. And then the vas deferens goes and connects with a bunch of different like accessory glands, like what is it called? The prostate gland, the bulbourethral gland, the the seminal vesicles, and all of these secrete like different things into the ejaculate. Um, I don't remember exactly what is in it. I just remember that there's vitamin C, right? The ascorbic acid, there are anticoagulants and coagulants. For some reason, there's both. Um, and fructose, I think, that's the nourishment for the sperm, right? I think sperm can survive like 2 days in the uterus. I guess that's how they survive. The female. Okay. Uh, ovaries, oviduct. Yeah, there's not much on the slide. So let's talk about the ovaries, I guess. So in each ovary, first of all, you have two ovaries, and you can see how they're attached by a ligament to this to the uterus, and in each ovary, it contains the follicles that give rise to egg cells. And a follicle is pretty much what it sounds like. It actually what is it called? It basically houses like a dormant female gamete, and every once a month, one of the follicles ruptures, releases releases a egg that goes through the oviduct into the ovary. And then, yeah. Here, I guess I can zoom here. Do you, do you guys see the zoom in? So you have, start from the bottom. You have the labia minora and majora, which are the two like folds or flaps. You have the vaginal opening, and the clitoris is like the what's it called? The not homologous. It's like a very similar structure to the male penis. Not vestigial. I can't find the word. But it has erectile tissue. It has the glands. It has the body. Yeah, and then yeah. Okay, I think that's enough of this. Gametogenesis. Okay. So how do these gametes actually arrive, right? Let's look at guys first. So sperm begin their development in the testes in the epididymis, right? So you can see here, this is the inside of a seminiferous tubule, or like inside the testes. And on the very outside, these are like germ cells. Not quite germ cells, but they're basically germ cells. And there are cells called Sertoli cells. And Sertoli cells, they're stimulated by FSH. And Sertoli cells nourish the developing sperm. Leydig cells, on the other hand, Leydig cells are stimulated by luteinizing hormone, and these produce testosterone, so androgens. So once again, Sertoli stimulated by FSH nourish the sperm. Leydig stimulated by luteinizing hormone secretes androgens, or male sex hormones. Right. All right. So the female. Oh, by the way, FSH and LH are like the. Oh God. The tropic hormones for your reproductive system. Like, for example, if you think of adrenocorticotropic hormone that stimulates your adrenal glands to grow, FSH and LH are the same for testes and ovaries, right? The female equivalent of Sertoli and Leydig cells are the granulosa and theca cells, and they're not exactly 100% analogous. So both granulosa and theca cells will nourish for the developing follicle. The difference, so that's the difference, right? For males, only Sertoli cells will nourish the sperm. Leydig cells only produce, Leydig cells, their function is to produce androgens, not to nourish the sperm. Female equivalent, granulosa and theca, they both nourish the sperm. They both nourish the gametes, and they both produce sex hormones. And I think granulosa produces androgens, and theca cells produce estrogens. So androgens are the male sex hormones. Estrogens are the female sex hormones. And I probably got them flipped. I'm not sure. But so female equivalents both nourish and produce hormones. And the ovarian and uterine / menstrual cycles, I think it's good to distinguish these two. So the menstrual cycle is like they're both 1 month long, but the menstrual cycle talks about like the the lining of the thickening of the lining of the uterus. I'm blanking so hard on what it's called. The thickening of the lining of the uterus, whereas the ovarian cycle depicts the changes, the fluctuations in hormones, the rupturing of follicles, the development of follicles. Yeah, so there's a diagram with the exact times in Campbell's if you're interested. I need to move on. All right, okay. Last chapter, animal development. Okay, so let's go back to fertilization. And we know that gametes are haploid, right? Gametes have one copy of the genome. Now, if you have one sperm and one egg, those two fuse and you get two copies of the genome, right? And then you have a diploid organism, which is perfect. That's what you want. But what happens if like let's say we're talking about frogs, and frogs spawn a bunch of eggs in the water, right? What happens if a sperm gets into an already fertilized egg? So now this egg has two sperm nuclei and one egg nuclei. So it has three copies of DNA. It's triploid now. And that if that happened, that egg would most likely fail. Excuse me? It would not be able to develop into a full organism. And so animals develop these blocks to polyspermy. Excuse me. Remember how I was talking about animals that spawn into water? They require they have more like specific requirements for sperm to enter like their eggs. They have more like safeguards against these kinds of things. Well, frogs have this thing called a fast. Have a fast block to polyspermy, and that means a fast block to polyspermy in general means that when a sperm immediately binds to an egg, that will immediately make the egg unviable to other sperm. So other sperm can no longer bind to that egg. And in frogs, this happens by the depolarization of the membrane. I'll go into a bit more detail on this. So frog eggs have like a vitelline, they have the cell membrane, they have a vitelline layer and a cell membrane, right? And on the, I think, actually, I might have gotten backwards, but on the outermost layer, it has like receptors, specific receptors for frog sperm. And when a frog sperm, what happens for when fertilization happens is a frog sperm will attach to the receptor. There will undergo a process called an acrosomal reaction, and eventually the nucleus gets injected into the egg cell. All right. Now, following the nucleus entering, immediately the cell membrane depolarizes, and all of the receptors fall off. Now, if all the receptors fall off, sperm can no longer recognize the egg, and therefore sperm nuclei cannot bind and go into the egg. Now that's a fast block, right? So once one sperm comes in, it gets rid of all the receptors, nothing else can come in. Now, what's the slow block? In frogs, there is a slow block that results in, so the outer layer of the egg like lifts outwards, right? It creates like a space and a bunch of calcium cortical granules. So these cortical vesicles filled with calcium like explode into that space and basically fill the the vitelline layer with a hard calcium covering. Now you have an egg that has a cell membrane and then a hard calcium like shell almost. And then inside is are the two, the sperm and the egg nuclei. And now even if a sperm were to randomly like wander into the nucleus, into the egg, it would get sought by the hard layer, right? And that's a slow block to polysperm. Okay, so now let's talk about cleavage. So once an egg is fertilized, the egg will undergo a process known as cleavage, where it's rapid cell division without increasing your cytoplasm, right? So it's rapid cell division without increasing your cytoplasm. So you end up with a bunch of smaller, tiny cells and a stage called a blastula. In frogs in particular, you have, because frog eggs have yolk, you separate, so you look at this egg, because frog eggs have yolk, they have what is known as the animal and the vegetal pole. So the animal pole has no yolk. That's its feature, right? The yellow, see this yellow? This is the vegetal pole. V for vegetal. And then this, the gray part, or not the gray, like the dark gray part, is A for animal, because it has no yolk. So the yolk is divided in half, or maybe a bit less than half, and the half with yolk is known as a vegetal pole. The half without yolk is known as the animal pole. Right. And so cleavage can either be holoblastic or meriblastic. And what does that mean? So holoblastic cleavage means that you're cutting complete your cleavage, like your cell division, the line of cell division goes completely through the egg, right? Like it splits the egg exactly in half. Meriblastic cell division, on the other hand, means the cleavage only goes like halfway or 3/4 way through. Basically, it doesn't cut the egg perfectly, like entirely in half. Right. And in the case of frogs, this happens because the animal pole divides normally, like just normal cell division. However, the vegetal pole has a lot of yolk, and because it has a lot of yolk, its cytoplasm is a lot more sticky. It's a lot more thick, and it's hard to cut through that thick layer. Because it's hard to cut through that thick layer, it divides more slowly, and therefore you end up with this uneven division where you have the blastocele, which is the cavity formed for the blastula. The blastocele is more on top than in the middle, right? So basically the smaller cells are on top, and the cells on the bottom are larger because cell division is slower. All right. And now the stage of the gastrula have different germ layers, and these are different from germ cells. Germ layers are like the starting layers, the embryonic layers of cells. You have ecto, meso, and endoderm. So ectoderm is outside, mesoderm is bones, connective tissue, muscle, yada yada, and endoderm is inside, like your lining your digestive tract, and a bunch of other organs. Now, animals can either be diplo or triploblast, and the difference between these two is whether or not they have mesoderm. Diploblast embryos only have ecto and endoderm, right? But triploblasts have all three. That's about it. Now let's talk about the amniotic egg. Okay, so 4 things that you should know. All right, one, look, ooh, cool, it's the embryo. Okay, next, the amnion. Amnion is a sac. It's the fluid-filled sac that cushions the embryo, where it gets its name from, right? Then, moving down, you have the allantoid and the chorion. So the allantoid and the chorion have similar functions, and they both aid in gas exchange and waste removal. Yeah, that's basically on allantoic chorion, gas exchange, and waste removal. And the yolk sac is like it's yolk. It's the food source. It's like proteins, the carbohydrates, whatever. Actually, I don't know if it's carbohydrates, but I okay. Last slide. I'll make this quick. Organogenesis, neurulation. So this is really cool. On the dorsal back to belly, dorsal ventral back to belly. On the dorsal layer of the embryo, of some animals, like for example, for frogs, a neural plate forms, and this neural plate will eventually fold in, as you see here, and that will form the neural tube. Neural tube becomes the spinal cord. Cell migration. So cells in embryos actually move around by like changing their shape, yada yada yada. Apoptosis, as you know, like for example, our fingers in embryo in utero are webbed. Apoptosis kills the webbing or the cells that make up the webbing so that our fingers look the way they are. Determination versus differentiation. Okay, everything after this is pretty specific. I'll just go over these two concepts and then we'll be done. Determination versus differentiation. So determination is it's very simple. Difference is very simple. So determination is basically I decide that I'm going to do this, right? I say I'm going to do this. Differentiation is actually the process of actually doing that. So for a cell, determination is saying I will become a muscle cell. Differentiation, it's doing the steps that actually make it a muscle cell. And finally, actually, out of context isn't that important. Okay, there we go. Thank you all for coming, and I'll see you next week. Take care, all.